Next, we are going to learn about active or adaptive immunotherapy, and it is an approach that tries to get a person's own immune system more involved in the fight against malignant cells. The immune system has evolved to recognize and destroy foreign materials, but cancer cells are derived from a person's own cells, although many tumor cells do contain proteins that are not expressed by their normal counterparts, so-called tumor-associated antigens or mutated proteins that are different from those present in normal cells. They are still basically host proteins present in host cells. As a result, the immune system typically fails to recognize these proteins as inappropriate, even if a person does possess immune cells that recognize certain tumor associated antigens. Tumors evoke a number of mechanisms that allow them to escape immune destruction. And many different strategies have been formulated to overcome these hurdles and artificially stimulate the immune system to mount a more vigorous response against tumor cells. One approach is to inoculate a patient with a protein not to be present in their cancer cells, such as HER2 in the case of a person suffering from breast cancer, or CEA, like carcinoembryonic antigen in a person with pancreatic cancer. If the body can be stimulated to mount an immune response against the protein, that response has the potential to attack the cancer cells that have that protein on their surface. In most immunotherapeutic approaches, immune cells are isolated from the patient, modified or stimulated in one way or another in vitro, allowing to proliferate in culture and then reintroduced into the patient. For many years, clinical trials using this strategy were disappointing, but recent publications have provided reasons for cautious optimism. In many of the studies, a significant minority of patients have exhibited a positive response to the treatment, which means that their tumors have at least shrunk in size or extent, and the patient's expected time of survival has increased significantly. And why some patients respond while most do not remains a focus of investigation. We will briefly consider two of the most promising examples of this general type of cell-based immunotherapy. Like, because of both the treatments to be described have to be personalized to the individual patient, they are likely to be extremely expensive should they ever become approved and widely available. And DCVAX is currently in phase 2 trials for the highly lethal brain tumor, glioblastoma. DCVAX utilizes dendritic cells that have been taken from the patient's blood, stimulated in vitro with antigens from the patient's own tumor, and then injected back into the patient's body. Because the strategy involves exposure of dendritic cells to specific proteins with the goal of generating an immune response, DCVAX is described as a cancer vaccine. The manufacturing process takes about 10 days and provides enough cells for several years of treatment. Although the number of patients that have been treated to date is limited to a few dozen, the vaccine appears to have extended the median survival time of patients to more than 3 years from time of diagnosis. Without such treatment, these patients would presumably have died in about 15 months. As of July 2010, 33% of treated patients had survived at least 4 years and 25% had survived at least 6 years. Fewer than 5% of patients given conventional treatment survive 5 years. In August 2011, the results of a phase 1 trial carried out on 3 patients with advanced chronic lymphatic leukemia, or CLL, was reported both in the literature and in the news media. Phase 1 trials are normally carried out to determine whether the drug is safe, its proper dosage. In this case, the treatment resulted in the complete remission of disease in 2 of patients and at least partial remission in the third. To carry out the treatment, Carl June and his colleagues at the University of Pennsylvania first isolated T cells from the blood of each of the patients, and T cells carry a multi-subunit protein on the surface of their cells called the T cell receptors, or TCR. The TCR determines which specific molecules or antigens that a given T cell will react with, and consequently which target cells that T cells will attack and likely kill. For the most part, T cells do not possess TCRs that would allow them to treat with other cells in the body, which protects the body from self-attack. In the clinical trial, researchers isolated T cells and genetically engineered these cells using a disabled HIV-1 viral vector to carry a specific high-affinity TCR that will that would allow T cells to react with and kill host cells that possess a CD19 protein on their surface. The only cells that normally carry CD19 are B cells, both the normal versions and those that are part of the CLL cancer. Once T cells are genetically modified, their numbers were greatly expanded in culture and were then infused back into the patient from which the T cells were originally obtained. Within a week or two, the patients were stricken with a sickness characterized by very high fever, lowered blood pressure, and kidney distress. The symptoms were the result of the war going on within their bodies between the infused T cell and the massive number of their progeny formed in the body after infusion, and the huge number of cancerous cells that these patients carried. It was estimated that each infused T cells was responsible for the death of more than 100 CLL cells. In the end, the patients were left with a relatively small number of genetically engineered T memory cells derived from the infusion, a virtual absence of normal B cells due to their destruction by the T cells, and either the absence of malignant cells or, in one case, a generally reduced tumor burden. The presence of memory T cells should prevent the re-emergence of CCL, as well as the cell would prevent the re-emergence of CLL, as well as the suppression of formation of normal B cells, which might make this patients more susceptible to infection. Keep in mind that the use of genetically modified T cells carries considerable risk because these T cells can attack and kill any cell in the body that happens to carry the targeted antigen. As a result, the types of cell-based immunotherapies have the potential to cause severe autoimmune side effects, and whether or not the approach used in the study can be safely applied to other types of cancer is an unanswered question. Then, inhibiting the activity of cancer promoting proteins, that is the next method. Cancer cells behave as they do because they contain proteins that are either present at normal concentrations or display abnormal activity. A number of these proteins were very important. In many cases, the growth or survival of tumor cells depend on the continued activity of one or more of these deviant proteins. This dependency is known as oncogene addiction. If the activity of one of these proteins can be selectively blocked, it should be possible to kill the entire population of malignant cells. With this goal in mind, researchers have synthesized a virtual arsenal of small molecular weight compounds that inhibit the activity of cancer promoting proteins. Some of these drugs were custom designed to inhibit a particular protein of known structure, where others were identified by randomly screening large number of compounds that have been synthesized by pharmaceutical companies. Once a protein inhibiting compound has been identified, it is typically tested for effectiveness against a panel of approximately 60 different types of cultured cells, each originally isolated from a different human cancer. Using a diverse panel of cells allows researchers to reconstitute the genetic heterogeneity and the diverse drug sensitivities found among human cancers. Success against cultured cells generally lead to tests of agent and immunocompromised mice carrying human tumor transplants or xenografts. Although numerous compounds in this list have shown some promise of halting the growth of various types of tumors, one compound has had unparalleled success in treating the patients with chronic myelogenous leukemia, CML. It was noted earlier that certain types of cancers are initiated by specific chromosome translocations, and CML is caused by a translocation that brings a proto-oncogene ABL in contact with another gene, BCR, to form a chimeric gene or BCR-ABL. Blood-forming cells that carry this translocation express a high level of ABL tyrosine kinase activity, which cause the cells to proliferate uncontrollably and indicate the process of tumorigenesis. Compound called Gleevec is identified and selectively inhibits ABL kinase by binding to the inactive form of protein and preventing its phosphorylation by another kinase, which is required for ABL activation. Initial clinical trials in Gleevec was remarkably successful, causing nearly all CML patients who recovered the drug at sufficiently high dose to go into remission. These studies confirmed the notion that elimination of a single required oncogene product could stop the growth of a human cancer. So, notion of elimination of a single required oncogene product could stop the growth of a human cancer. The drug was rapidly approved and has been in use for years. Patients have to continue to take the drug to remain in remission, and many of them, especially those who began treatment as an advanced stage, eventually develop drug resistance. Oh no. So, most cases of resistance result from mutations in the ABL portion of the fusion gene. This has prompted the development of a second generation of target inhibitors that remain active against most of the mutated forms of ABL kinase. These drugs appear to be effective in treating Gleevec-resistant cases of CML and suggest that the ideal drug regimen may consist of a cocktail of several different inhibitors that target different parts of the same protein, ensuring that drug-resistant mutants will not emerge. Then it was hoped that Gleevec would be followed rapidly by numerous other highly effective protein inhibiting drugs. Although a number of protein targeting small molecule inhibitors have shown modest success in clinical trials and have been approved by the FDA, the hundreds of others are being tested in the clinic. None of them studied to date have been able to fully stop the growth of any of these common solid cancers, namely those of the breast, lung, prostate, or pancreas. One major success has been achieved. A drug called Zelboraf has recently been approved for the treatment of patients with metastatic melanoma whose cancer is driven by a specific mutated version of BRAF1 in which the normal valine residue at position 600 is replaced by the glutamic acid BRAF V600E, thereby changing the shape of the active site. This mutation occurs in approximately 50% of melanoma patients, and Zelboraf specifically blocks the activity of this mutant enzyme without affecting the normal protein. As with Gleevec, patients develop resistance to Zelboraf, but unlike Gleevec, resistance appears much earlier, typically after 7 months of treatment, and in most cases results from mutations in other genes rather than secondary mutations in BRAF. Resistance to Zelboraf typically occurs because of MAPK pathway once again become constitutively active, bypassing the cell's need for BRAF kinase activity, which continues to be inhibited by the drug. Studies are underway to combine Zelboraf with inhibitors of downstream components of the pathway such as MAPK or ERK. It is unlikely that a tumor will harbor cells that are resistant to two drugs that target two different pathways or two different proteins in the same pathway. Consequently, an approach that uses a combination of drugs may be the best way to block the emergence of resistant cells. Inhibiting the reason for the failure to develop drugs that can irradiate solid epithelial based tumors or carcinomas are not entirely clear. One reason may be that these tumors are more complex genetically and the cells are not as dependent on a single oncogene product and aberrant signaling pathways as the blood cell cancers and melanoma. Another reason may be that only a fraction of the patients with a particular type of tumor are sensitive to a particular drug. This was originally suggested in studies of Eracia, an inhibitor of tyrosine kinase of the EGF receptor. Eracia is originally tested on lung cancer patients because these tumors were known to exhibit high levels of EGFR. Initial clinical trials found that approximately 10% of patients in the United States and 30% of Japanese patients responded progressively and positively to the drug, where the remaining population was unaffected. Subsequent studies on cancers that contained EGFR mutations indicate that targeting the mutant EGFR was only potentially successful if the patient had wild type KRAS. Those patients whose cancers also exhibited a mutant KRAS gene were non-responders. As a result, lung and colon cancers are now tested for KRAS mutations before anti-EGFR therapy is initiated. Most recently, it was discovered that the drug Xalkori is highly effective in treating a select subgroup of lung cancer patients that had normal EGFR gene but expressed an EML4ALK fusion protein that results in a chromosomal translocation. Then Xalkori inhibits ALK kinase. The drug was approved along with a companion diagnostic test that uses fluorescence sensitive hybridization of fish to identify the presence of a rearrangement, which occurs in about 5% of patients. These findings verify the notion that not all cases of particular cancer can be treated alike. Instead, targeted cancer therapies will ultimately have to be tailored to fit the specific genetic modifications present in the tumors of individual patients. With this in mind, a number of major cancer centers have begun programs to sequence a panel of selected genes in patients with certain cancers. This information is also being able to help select individuals for participation in clinical trials, thereby increasing the likelihood of finding drugs that are effective in treating subsets of patients with a certain type of cancer. Without this type of genetic screening, the positive signals for a drug that might be useful for a subset of patients can be drawn out by the lack of clinical benefits to the population as a whole. The current challenge is to identify genetic markers that have value in predicting the success or failure of a given therapy. Then we have focused on this section upon targeting therapies that inhibit proteins that are either abnormal themselves or the abnormally expressed in cancer cells, but there may be also be proteins that have a normal structure and expression yet for some reason play an important role in the lives of cancer cells. Inhibitors that target such proteins might have considerable potential as therapeutic agents in the treatment of cancer. We have seen in this chapter how cancer cells contain mutations in a wide variety of genes, many of which lead to the overall inhibition of certain metabolic or signaling pathways. While this may help promote the growth and survival of these cancer cells, it may also cause them to become more independent than normal cells upon their pathway that continue to operate in normal fashion. Recent announcements hailing the promise of PARP1 inhibitors in the treatment of several types of cancer provide an example of this type of reasoning. PARP, which is an acronym for poly-ADP ribose polymerase, is a poorly understood enzyme that is involved in numerous processes involving DNA metabolism, including DNA repair. PARP1 inhibitors have shown particular promise in the treatment of breast and ovarian cancers that exhibit deficiencies in BRCA1 or BRCA2. BRCA proteins are involved in DNA repair, and it is reasoned that such cancer cells are more dependent on normal cells upon other DNA repair pathways, including those that require PRP1. When PRP1 is inhibited in BRCA deficient tumor cells, certain types of DNA damage cannot be repaired, causing the cells to die by apoptosis. This type of treatment is based on a strategy of synthetic lethality, which suggests that mutations or inhibition of any one protein has no effect on cell viability, but that mutation and or inhibition in two different proteins leave the cell unable to carry out one or more essential functions. More importantly, this approach provides general strategies to target cancer cells that have lost the function of a particular tumor suppressor protein, and in this case BRCA1 or BRCA2. A similar strategy might also be devised to target cancer cells that have lost p53 function, for example, because such cells should also be vulnerable to drugs that have no effect on normal cells which possess intact tumor suppressor pathways. It might be possible to find genes that might be sensitive to synthetic lethality by searching the data from cancer genomic studies and looking for either two genes that are never mutated together in the same tumor or genes that are never mutated in any case of particular cancer. Genes in this later category are presumably required for survival of the cancer cells and represent possible drug targets. Then the concept of a cancer stem cell. Another reason for the failure to develop more effective targeted therapies may be that the agents that are not targeting the appropriate cells within the tumor. This possibly requires further explanation, but raises an important issue concerning both the basic biology of cancer and its treatment. Throughout this chapter, we have considered a tumor to be a mass of relatively homogeneous cells. When viewed in this way, all of the cells in the tumor are capable of unlimited proliferation, and all of the cells have the ability and opportunity to evolve into a more malignant phenotype as the result of ongoing genetic change. In recent years, a new concept has emerged, which suggests that while most of the cells of a tumor may be dividing at a rapid rate, they have relatively limited long term potential to sustain the primary tumor or indicate or initiate a new secondary tumor. Instead, a relatively small fraction of cells scattered throughout the tumor are responsible for maintaining the tumor and promoting its spread. The special cells are known as cancer stem cells, and there is considerable experimental evidence for their existence in leukemias, brain tumors, breast tumors, and other cancers. In most cases, the evidence for the existence of cancer stem cells rests on the finding that only a subpopulation of cells in a given human tumor is able to initiate the formation of a new tumor following injection into a susceptible or immunodeficient mouse. From cells that are able to regrow the tumor in the inoculated animal often possesses certain surface proteins that distinguish them from the cells that make up the bulk of the tumor, and it is these makers that serve to define the cancer stem cells. The term cancer stem cell does not suggest anything about the origin of the cells, whether any are tissue or stem cell or some other type, nor does it imply that cancer stem cells necessarily resemble normal tissue stem cells. Instead, they are defined simply as the cells capable of propagating or regrowing the tumor. The concept of the cancer stem cells is raised at this point in the chapter because it has important consequences for cancer therapy. If it is true that only a small subpopulation of cells in the tumor has the capability of continuing the life of that tumor, then developing drugs that rapidly kill off that bulk of tumor mass but spare the cancer stem cells is doomed and ultimately to fail. This is presumably the reason that patients with CML might continue to take Gleevec to keep their diseases in remission. Gleevec does not seem to kill the leukemia initiating cells responsible for the occurrence of a disease, but there are efforts underway to identify cancer stem cells in various types of tumors and learn more about their properties. These new views are just beginning to have an impact on drug development. Then next topic, inhibiting the formation of new blood vessels or angiogenesis. As a tumor grows in size, it stimulates the formation of new blood vessels, a process called angiogenesis. Blood vessels are required to deliver nutrients and oxygen to the rapidly growing tumor cells and to remove their waste products. Blood vessels also provide the conduits of cancer cells to spread to other sites in the body. In 1971, Judah Folkman and of Harvard University suggested that solid tumors might be destroyed by inhibiting their ability to form new blood vessels. After a quarter of century of relative obscurity, this idea has been translated into an approved anti cancer strategy. Cancer cells promote angiogenesis by secreting growth factors such as VEGF that act on the endothelial cells of surrounding blood vessels, stimulating them to proliferate and develop into new vessels. A variety of angiogenic inhibitors have been developed by biotechnology companies, including antibodies and synthetic compounds directing against integrins, and growth factor receptors. Preclinical studies on mice and rats suggested that angiogenic inhibitors might be effective in stopping tumor growth. Most importantly, tumor treated in this way of these inhibitors in preclinical studies did not become resistant to repeated drug application. Tumor cells become resistant to the usual chemotherapeutic agents because the cells are genetically unstable and can evolve into resistant forms. And inhibitors of angiogenesis, however, target normal genetically stable endothelial cells which continue to respond to the presence of these agents. Then, inhibiting angiogenesis in human tumors is not as easily accomplished as might have been expected based on studies with mice. To date, the most promising results have been obtained with humanized antibody called Avastin that is directed against VEGF, endothelial cell growth factor that is overexpressed in most solid tumors. Avastin blocks VEGF from binding to and activating its receptor like VEGFR. FAD approval has been based on clinical trials demonstrating that Avastin, the combination of both standard chemotherapy, could prolong the lives of patients with metastatic colorectal cancer for several months. Subsequent studies suggest that Avastin could add several months to live the patients with several other types of solid tumors, but this conclusion has been debated, most notably in cases of breast cancer. Whether or not inhibition of angiogenesis will ever prove itself as an effective therapy remains to be seen, but at the present time, interest in this therapeutic strategy has waned. For the foreseeable future, the best anti cancer strategy is early detection. The time that elapses between the initiation of one of the common endothelial cancers or epithelial cancers and its progression to a deadly metastatic disease is generally indicated to be an at least a decade and often much longer. Consequently, there is ample time to detect these cancers before they become life threatening. A number of screening procedures are currently in use, including mammography for detecting breast cancer, pap smears for detecting cervical cancer, PSA determinations for detecting prostate cancer, and colonoscopy for detecting colorectal cancer. It is hoped that advances in proteomics will eventually lead to the development of new screening tests based on the relative levels of various proteins present in blood or other bodily fluids. This approach was discussed later, and some blood-borne biological indicators or biomarkers that would reveal the presence of cancer, including mutant DNA, particular miRNA, abnormal carbohydrates, and distinctive metabolites. Screening tests based on each of these types of biomarkers are being investigated. Now let's have a quick summary of this all major points discussed in this chapter. Like we will be having a short synopsis. So, cancer is a disease involving heritable defects in cellular control mechanisms that result in the formation of invasive tumors capable of releasing cells that can spread the disease to distinct sites in the body. Many of the characteristics of tumor cells can be observed in culture, whereas normal cells proliferate until they form a single layer or monolayer over the bottom of the dish. Cancer cells continue to grow in culture, piling on top of the another to form clumps. Other characteristics often revealed by cancer cells include an abnormal number of chromosomes, an ability to continue divide indefinitely, a dependence on glycolysis, and lack of responsiveness to neighboring cells. Then normal cells can be converted to cancer cells by treatment with a wide variety of chemicals, ionizing radiations, and a variety of DNA and RNA containing viruses. All of these agents act by causing changes in the genome of the transformed cell. Analysis of the cells of cancerous tumors almost always shows that the cells have arisen by growth of a single cell. A tumor is said to be monoclonal. The development of a malignant tumor is a multi-step process characterized by a progression of genetic alterations that make the cells increasingly less responsive to the body's normal regulatory machinery and better able to invade normal tissue. The genes involved in carcinogenesis constitute a specific subset of genome whose products are involved in such activities as control of the cell cycle, intercellular adhesion, and DNA repair. The level of expression of particular genes in different parts of cancers can be determined using DNA microarrays in addition to genetic alterations. Growth of tumor protein cells are also influenced by non-genetic and epigenetic influences that allow the cells to express its malignant phenotype.